You got kids.
I have a 3 and 5-year-old.
Oh, Lordy, they listened to you, I'm sure. Okay. All right. So I'd like to introduce today Dr. Alvin Thomas. He's a postdoctoral fellow at the School of Medicine, and he leverages multi-omic, psychometric, and brain imaging data to understand the biology of cognitive resilience to Alzheimer's disease and related disorders. Very impressive.
Well done.
And Dr. Thomas's other interests include outcomes research in kidney and liver transplantation and methodologic work in deep learning. He earned a BS with honors in chemistry and engineering from Washington and Lee University, an MS-PH in global disease epi from Johns Hopkins, and a PhD in epi from University of North Carolina at Chapel Hill, which we all know is not a very good school.
What are you talking about?
I knew you were there, and I knew Rachel was here. I did that on purpose, yes. See if you were awake, Matt. He served in the AmeriCorps program and is committed to diminishing poverty through research and service, and we're very pleased to have him here. So I'm going to turn this over to you and see what you have to say.
Thank you. So today's a big day for public health here, not because of the snow, but because we had our assembly. Miguel Hernan speaking right after me. So in the interest of time, I've cut a few slides so that people could get over there if they want. But anyway, I'm very excited to use this time to talk a little bit about studying diseases associated with aging and some of my work in frailty and resilience, and hopefully get you excited about the issue of aging and what we can do to fix it. I have nothing to disclose in terms of financial conflicts, but I would like to acknowledge the NIH for funding this and other very important work. It would not be possible to do this kind of research without NIH support. So I would have started with why health, but Sandra actually gave the prequel to my talk a few weeks ago. So instead, I'll just share one of his slides and make the point or reiterate the point that health ultimately is about allowing people to flourish, to live the best life that they can. It is the reason I think a lot about health span instead of lifespan. And especially in the context of aging, this is a critical idea to kind of ingrain in your being. And when we think about aging and we think about health, you often think about these frameworks where, as you see in panel A, you have these trajectories and people are on these healthy trajectories or at risk or at these poor marginal trajectories of health. The reality is probably a little bit closer to what we see in panel B. And the most important thing for us in academia and practice in public health is that these trajectories are modifiable. There are things that we can do, there are things that people do, there's things that society does that can increase or decrease your overall health. And that's true regardless of your phase of life. But today I'm going to be talking mostly about late life. Part of the reason for this, why it's important, is that the world is aging. There's wonderful work in demography that shows in very clear pictures that I'm not going to show today, that most populations in most countries are starting to invert in terms of their age distribution in both sexes. And with that is this increase in the absolute number of individuals over the age of 65, which for the purposes of this talk we'll call older adult, but that's just a number. So it's both an increase in the proportion and in the absolute number. These are both critical to think about when we think about our already stressed health system and our even more stressed public health system. Beyond that, of course, aging is associated with disease risk. And not only is it associated with disease risk in many parts of the world, it will be the primary driver of increases in the global burden of disease. The arrow is pointing to high-income North America, and you see that lighter green is the contribution of aging to this increase in risk in the global burden of disease from the Global Burden of Disease publication in 2022. The darker green, by the way, is just population growth. So in some parts of the world over the next few decades, population growth is going to be a major driver, but everyone is going to shift in this direction based on what we've learned from our colleagues in demography. I'm going to go into a little bit more detail later on, but I just want to say something about chronologic health or chronologic aging and biological aging. This may be a familiar topic to some people. It may not be. When I'm talking about aging, most often in this context, I will be talking about just time, chronologic aging, but the driver from a biology point of view is actually happening at the molecular cellular level. Something is going on. We really don't know. We've had a lot of guesses and a lot of different frameworks to think about it. But the consequence, at least, is that cells stop doing some of their critical functions in DNA repair, in addressing somatic mutations, and ultimately just getting rid of cells that aren't doing what they need to be doing. This is why we start losing function in all our organs and so on. This is ultimately what leads to disease, or at least the increased risk of disease for the things I'll be talking about today. As an epidemiologist, I think a lot about study design and asking the right question with the right study. Aging is a very challenging space to work in. One, because age is common. We're all exposed to it. So it's difficult to think about when an exposure starts and ends, how the very framework of like a cohort study would work. There are consequences of survivorship that actually may be important to some questions and are not even coming to the forefront when the study is designed. And there are cohort effects. Right now, if you're studying baby boomers, there are things that they lived through that I have not lived through that could have profound effects on health. And the questions that I'm asking and the answers I'm getting may not actually apply, at least in terms of the actual estimate, to me or to others in the room, or to people that already have lived and died and who we have data for. So these challenges are difficult to surmount. There are a lot of solutions. One way to approach it is start thinking about individuals and their level of vulnerability. There's a lot of different ways to phrase this. One context is thinking about frailty or increased vulnerability to disease, and resilience, which is an decreased vulnerability. So in this context, even though we're interested in population health, population estimates, that's the level of inference we're often making, we're in the moment thinking about the individual and what are the factors that make this individual at increased risk or at lower risk, which is actually a much harder question, as we'll come to later on in the presentation. Now, to address this, I'm going to talk about two different bodies of work, one in kidney disease and one in dementia. For the kidney disease part of this talk, we're going to look at frailty as a model. Kidney disease is prevalent. 10% of the population are estimated to have kidney disease, which is just an absurd number. If you think globally, that's 800,000 people that are somewhere along the kidney disease spectrum. The optimal treatment is a kidney transplant. Actually, a living donor kidney transplant would be the best thing. It could give you an additional 20 years of life or more. And that would be wonderful if we could do it, but we can't. There are not enough living donors. There aren't enough deceased donors. And some of my colleagues are working right now in xenotransplant, and I have others that work in trying to create organs from scratch, but we're just not there yet. So right now we have this very large population of individuals with chronic kidney disease, some who need to go on dialysis, which is in a very exhausting process that comes with a lot of risks on its own, and those that reach the final stages, end stage kidney disease, have a very high risk of mortality and live with a lot of morbidity if they're not able to get a transplant. This work we'll talk about, we're pulling data from two different studies. One is actually the National Registry. In the United States, we have a registry that has all transplants for kidney, liver, heart, lung, and some of our soft tissues as well since 1987. It's a wonderful resource as an epidemiologist. You have the full study, the full source, target population, it's all there. But it doesn't go into as much detail as you need to answer some of the questions you're interested in, especially questions that may not relate directly to allocation, which is why the registry was created in the first place. So we also use data from this multi-site study called FAIR, which is the Frailty Assessment in Renal Disease study. And in this study, we recruited older adults that were transplant candidates, and they underwent cognitive testing, a lot of other sort of physical testing and assessments. And it allows us to ask questions now in the context of this older age population about their risks and outcomes with kidney disease. Another nice thing, just as a small point, when you think about the transplant procedure itself, it's actually a very clean time origin. So if you love time to effect models, if you love just having a nice cohort assembled, you can align everybody by the procedure. So that's also very nice. So these are two studies that I did early in my public health research life. The first one had to do with impairments and outcomes after kidney transplantation. So one thing that is not very well discussed is how individuals that have end-stage renal disease don't or do get listed for a kidney transplant. There has to be a feeling among the committee, which is composed of doctors, nurses, social workers, other professionals, that this kidney, which is a scarce resource that this person might receive, will work in them and lengthen their life. And there are going to be some individuals that are more vulnerable, that are less likely to live many years, and there are some that are maybe young and could live very long. So this is a very challenging conversation to have. There's obviously national rules around this, but there's a lot that happen in these rooms that can be kind of hidden from the public. And one of them is around impairments and how we think about it. This was something, actually, there were no papers on it at the time when we started this work. And our question was around visual impairments, hearing impairments, and mobility impairments, primarily. How frequent or how prevalent is it? You know, is it common in the candidate pool? Is it common in recipients? Are there any adverse outcomes? Are there special considerations we need to take? What we found in our study was actually a pretty high prevalence, which isn't necessarily surprising, especially for the visual side and the mobility side, because diabetes and kidney disease are very closely linked together. But what wasn't quite expected was the increased risk that these individuals had for mortality or for graft loss after they received the transplant. Their hazard ratio was 3 times higher than individuals that were otherwise similar except for the impairment. We didn't quite know what to do with this data, because on one hand, it is great that these individuals were able to get a transplant and live extra years of life, but this increased risk is something that needs to be addressed. And it was actually a few years later where there This study and another study that was published in JAMA like a few years after ours was the motivation for this policy directive from the American Medical Association around equitable access to transplantation. So our bit about impairments was actually removed from that language, but the purpose of this directive was to open the conversation around disability, the finalized policies around intellectual disabilities and developmental disabilities, but also in our minds, mobility disabilities and how that is being looked at systematically in these decisions. It's not an easy case to make on either side, but there wasn't a conversation going on, and we were very glad that our research helped get this conversation going. The second paper looked at cognitive impairments, and this actually became then the inspiration for all my work in dementia. Dementia is a contraindication for transplantation. So if you have dementia, you're not going to get listed for a kidney transplant. But dementia has a very long prodromal period, decades, and throughout that process, some individuals might experience mild to like more severe, moderate levels of impairment, and they are eligible for transplantation. We wanted to understand how prevalent it was again, and if there were any risks. And we did find that it was relatively common, that about 10% had some level of impairment. And in terms of absolute risk, there was a staggering 30% risk difference in their 5-year risk of all-cause graft loss and mortality. This actually was cited by the EU in their latest guidelines around the process of managing chronic kidney disease and transplantation. And their argument based on our work and others is that early diagnosis and clinical management is critical in the chronic kidney disease space, that time, because if you wait too long and these impairments persist, their benefits of transplantation are decreased, as is their access. And this actually is the seed of why I stopped working on frailty. Ultimately, what this work did, I think it was very important, but it was also a bit sad that by the time we were there to look at these outcomes and to look at these individuals, they were at a point where not a lot could be done. And I think the EU's recommendation in this regard is right. As a public health researcher, the most critical outcome was that people now are thinking about this earlier and looking at the early stages of chronic kidney disease, because more and more research is coming out about the connection between the kidney and the brain and how those on dialysis really need to think about cognitive impairments and monitor for potential dementia as well. So I just to summarize this section, this approach, looking at frailty, looking at factors that increase vulnerability, is quite common, and it is quite useful. As a huge field, we've learned a lot about risk factors and individual vulnerability and the impact that exposures can have on outcomes. But often the message is that you have to do things early, regardless of the disease you're studying. You have to move earlier in the pipeline and try to do preventative measures in order to make a big difference at the population level. For kidney disease, this is really important because these individuals actually exhibit accelerated aging. At the cellular level, once you have kidney disease, your metabolism is shot and your organs are aging faster than they would otherwise. And so you start accumulating all these other comorbidities. And that's why when you get to end-stage renal disease, it becomes very difficult to do anything other than give you a new kidney. So after this work, and especially inspired by the cognitive impairment work, I started thinking a lot about the brain and thinking a lot about dementia. And this in part is motivated by the lack of real treatments for dementia. At least in the kidney transplant space, there's dialysis, which could keep you alive for many years, even if it's exhausting. And then there's the transplant, which could give you a whole new chance at life. But with dementia, there isn't a lot that we have, including our latest anti-amyloid treatments. The efficacy in terms of how long it might prolong normal cognitive thinking is not where we'd want it to be. It's better than nothing. It's important that we have it, but again, we have to be working much earlier in the pipeline, because once the time dementia comes, there's right now not a lot that we can do. So dementia is a progressive decline in cognition. Alzheimer's disease is the most common cause of it, and the stereotypical biomarkers associated with Alzheimer's are amyloid, plaques, and tau tangles. It's on the rise. Of course, this is to be expected given our earlier discussion about demographics and risk factors. So it is the total number of cases are increasing, but with that is also the cost of caring for them. And the new drugs have only made it a little bit harder to make the argument for treating these individuals that are struggling with dementia. So putting on my public health hat, thinking now, this was in my early years of the PhD, what do I want to do with my research? What am I hoping to impact? I thought a lot about this finding. So when you think about biomarkers for Alzheimer's disease, you have on this graph, by the way, age is increasing on the x-axis, and on the y-axis is abnormalities. So the first thing you have is something like amyloid beta in your cerebral spinal fluid. Everyone sees some increase over time with age. And then you have these other sort of biomarkers that come into the picture. But it's only after you reach a certain point that cognition is actually impacted, right? So everyone is aging, you're getting these biomarkers building up, but that doesn't really mean anything until some point where the dam breaks and you start seeing impacts in your cognition. So if you look at any point in time, which would be a vertical line, you can see someone's level of cognition and then their biomarker profile. This is how a lot of work is done. And if you're thinking from a frailty perspective, you might be interested in those that are at increased risk, maybe, of these biomarkers, right? Or the change in cognition. But you can also look at the other side. There are some people that have the exact same biomarker profile. If you looked at their brain, they would have a lot of neuronal loss. There are actual holes in their brain, but you would have no idea looking at them. And this has been known for over 100 years. There's a huge discussion in Germany in the 1930s, 1940s, about whether Alzheimer, the doctor, was actually wrong with his discussion about amyloid plaques, because there are all these people that have amyloid plaques and don't have dementia. They didn't exhibit any of these cases. So those people lost out, but it took about 60 years for that pathologic finding to be brought back up in the psychology literature to think about, wait a second, we see this biological change, but these individuals are cognitively intact. And there are a lot of mechanisms that have been proposed to explain this. And as a field, we're very much in conflict about what's right and what's appropriate in terms of terminology. But what we've come to is that you can use resilience as a general term to kind of encompass this whole umbrella of potential mechanisms. And what we mean by resilience is really coming back to this picture. For a given profile of the brain or other biomarkers, do you have more cognitive function than we expect or less? And if you're on the more side, we call you cognitively resilient. So I wanted to study cognitive resilience and understand more about it, not just, is it protective, is it useful, but can we understand something about the biology? So this data comes from the ERIC study, which may be familiar to most people in the room. Very briefly, started off as a cardiovascular study, and as many cardiovascular studies have, become a study of aging, because that's a great way to utilize the amazing data that these individuals have given us over the last 30 years. During the 5th visit, there was a sub-study that did brain imaging with MRI. So we have about 2,000 participants, and what I did was create this measure of cognitive resilience shown by the equation at the bottom. So generally what we're doing is we're taking a measure of brain health, which is going to come from MRI data. So if you think about this in more detail, really what I'm talking about is there atrophy or not? How much loss of actual neurons of brain mass has happened? Because again, in general, as you age, you're going to lose brain cells, and they're some of the only cells that don't really regenerate. The outcome is cognition. Again, we're trying to find are those individuals that have higher cognition than we would expect, than we would predict. So for that, I used 10 psychometric tests, and we used my colleague Alvin Gross at Hopkins published on this a while ago. But there's a lot of different ways to come up with this general cognitive factor score. The idea is we wanted a cognitive score that wasn't specific to memory or specific to functioning. We wanted to just have a general score that would be sensitive to various changes, because again, with dementia, and not all of it is going to be Alzheimer's, some things go before others, and that varies by individual. On the MRI side, we ultimately pulled 20 features from the MRI data. We decided to do this a priori, but I did also create a few machine learning and deep learning models to do some selection. Ultimately, there wasn't a lot of difference between them, because what we've learned, thankfully, we've learned something over the past few decades, is where atrophy starts, right? So that was already captured by our pre-specification. So we were looking at the temporal and parietal lobes of the brain, looking at regions like the hippocampus, which is associated with memory, and those were the strong signals that also came out of the machine learning models. Importantly, we also include vascular lesions. And I say this because the ERIC study recruited both white and African-American participants. And from our work in the ERIC study and elsewhere, vascular presentations are actually more common in African-American individuals, often underlooked. And it creates these strange findings that aren't strange when you look at the whole picture about, risk of dementia and Black versus white individuals, and how much amyloid you need to see this change in cognition. When you look at the whole brain and you think about also the vascular impacts, this kind of evens out. But this was not previously included in studies that take a similar approach. Most people don't have a lot of great vascular data, but we did have that, and we also had Black participants. So we wanted to make sure that they were represented appropriately. So we derived this measure of cognitive resilience. I'm not going to go into the details, but I'm happy to if people want to discuss it. And the first thing we did is just validate the measure, because this is the first time anyone had done it in our cohort. There's a ton of data here. I'm not going to go into much detail, just briefly going to say the first thing we did is we looked at, is cognitive resilience associated with the things that it should be according to theory? And is it not associated with the things that it shouldn't be, again, according to theory? And that did end up being true. So ApoE4, which is the strongest genetic risk factor for Alzheimer's disease, is actually not associated with cognitive resilience. I think this was one of those moments where I finally said, thank goodness. Because when I started working in this space, the biggest question I had is, aren't the protective things just the opposite of the things that make you at risk, right? So if cardiovascular disease increases your risk, not having cardiovascular disease is protective, right? Well, that's not really true. There's actually more of a distance between states of health and states of disease. And we actually knew some of this from the genetic work, but it also pops out here and in other studies, where if you look at the genetic risk of Alzheimer's, it's not associated with resilience. There's different mechanisms at play here that it's actually quite exciting that this came out, because now you actually see the potential for new mechanisms of new solutions, new interventions that could work. Things like education, long established as a protective factor, highly associated with our measure of cognitive resilience. Exercise associated with cognitive resilience. Things that are negatively associated are depression and ultimately their cognitive score. We looked at the association between cognitive resilience and incident dementia in our participants. And we also looked at mortality. And again, just briefly, we did see the protective effect that we anticipated. So these are adults in their 60s, 70s, 80s. So we expect, of course, some level of mortality and incident dementia. And those with higher levels of cognitive resilience had about a 12% lower risk of mortality, 17% lower risk of incident dementia. But this did vary by race and sex. Now, I don't really believe we have the sample size to get the most accurate measure of this effect modification, but I do think there's something to it, especially on this in regards to cognitive resilience in women versus men. That has been well discussed, I think, especially in the genetics literature. But we might also be seeing something related to socially constructed race, and that's worth digging into. So briefly from this section, what I did, then this paper is under review right now, but I'm happy to share it with anyone. We created a measure of cognitive resilience. We adapted it to our population, which included African-American individuals, and therefore we wanted to include a better picture of brain health, including vascular factors. And we did ultimately validate our measure and find potentially this novel interaction between race, sex, and risk of dementia and mortality. But now that we had this measure, we moved to our main goal, which was to understand or see if we could understand a little bit more about the biology of cognitive resilience. So very briefly, a subset of the population from the first study had proteomics data in blood. And so we did an analysis to identify proteins that might be associated with cognitive resilience in this population, and some follow-up work to kind of dig into those relationships. So we did identify 30 proteins that were associated with cognitive resilience. Some were increased in those with cognitive resilience, and some were decreased, of course. And a few were only identified because of our sex-race interaction term, which I won't go into detail here, but there are some pathways, especially along the inflammation pathways, that might be more strongly associated in the African-American participants in our study relative to the white participants. But again, sample size was limited, so we take those findings with a grain of salt. Once we had these proteins, we looked at molecular networks, we looked at canonical pathways, we tried to dig into the biology as best as we could. And again, this kind of study in epidemiology is best for hypothesis generation, right? Most of what we're doing here needs to be followed up in the lab, and it's fun to work with our friends on the bench side and try to understand what could be going on. But it can also do things like identify druggable targets that we as epidemiologists could use pharmacoepi and things like that to look at as well. So one of the pathways that's shown here uses PI3K, and as a central element of this molecular network. This actually has been previously identified in like 20 years ago as a neuroprotective pathway and in the brain associated with neuronal survival, synaptic plasticity, myelination, which was kind of wonderful to see that this was actually the most dominant network predicted by the proteins that we identified. Other things that came up include a very strong involvement of the immune system. And there's now a lot of studies that are really leaning into this hypothesis around the importance of the immune system, whether Alzheimer's is actually an autoimmune disease, things in that way. So we did actually find a lot of signals around the proper regulation of the immune response, specifically that the initial immune response is kind of quelled appropriately. So those are the kind of proteins that were elevated. And among the pathways that we found that were interesting is this one that was discovered like 30 years ago, but then never talked about again, which is DHCR24. And it's interesting because actually it's come up now in a few colleagues have actually identified this again in some recent work. So it might be emerging or re-emerging. And this is a critical pathway in cholesterol biosynthesis. And again, that connects to, in the biology, right, the myelination. So basically how well signals are passing in the brain, and especially brain to the periphery, the rest of the body. So in summary, from this section, and again, I'm happy to go into more details, but we were able to take this measure of cognitive resilience that we developed and actually make novel insights into the actual biology of disease. And this is exciting, not just because these are potential pathways for health, but also potential pathways to protect against or to actually address Alzheimer's disease itself. And I think that's one of the positive things about looking at resilience versus frailty, is that potential dual impact that you could have. So in summary, what I hoped to do today is just start the conversation about population aging and how we could study it as epidemiologists. Again, there are many other frameworks, but what I discussed today are frameworks that look at individual vulnerability. What makes an individual at higher vulnerability or lower vulnerability, and how can we ultimately, in my opinion, promote resilience in the population? How can we help individuals avoid things like amyloid deposition would be great, but the reality is it's probably going to happen anyway. So once it does happen, how do we make sure that they remain cognitively intact? How do we make sure that they can live the life that they want to live, which is the purpose of health? So there are things we can learn from both models, but I do have a bias. And I would like to thank all my colleagues who have supported this work over the past decade, including my PhD mentor, Gerardo Heiss, who unfortunately passed away during my PhD, complications of COVID. But he and I were just sitting for years together, just the two of us working on this idea. That's where all the resilience work came from. And I think it's just wonderful that I now have the opportunity to share it. As well as my co-authors here. And of course, I did want to acknowledge some funding from the Reserve and Resilience Workshop and Melodem Consortium that have helped me refine some of these ideas, go to conferences, things like that. So with that, I'd like to thank you for your attention, and I'd be happy to answer any questions. Thank you. Yeah.
So in the resilience model that you shared in the last study, you know, you're limited to what data were collected in the cohorts.
Mm-hmm.
If you had if you had control over it and you could imagine like 3 things you would have loved to have had data about, what might those have been?
It's a really great question, because I thought I knew the answer, And about 3 months ago, I started graphing some things out and realized that actually is going to make it much harder. So my original answer was going to be something like fMRI, functional magnetic resonance imaging. You get a sense of which regions of the brain are activating. You get a brain metabolism, which is this core hypothesis to resilience, right? Like that you're actually seeing, lower metabolism and things like that, or in the case of resilience, higher metabolism. And one of the things I realized as I was modeling this out is that it's fine if you're thinking about totally normal controls, which normal, I don't really know what it means, and then dementia. But there's this funny thing that happens in between there during this mild cognitive impairment phase that I think is very underappreciated. And what some of the fMRI studies have shown us is that actually these like biomarkers and from the fMRI, the abnormalities that you see, they're kind of increasing for a period of time as you would expect, and then they're decreasing, but then they actually start increasing. So you expect the function of the brain basically to decrease as cognition is decreasing. But instead, what happens is the brain fights back during this mild cognitive impairment phase. Your brain is trying really hard to compensate, and then the dam breaks. Something stops, and then it's downhill. All the function is downhill, everything like that. But it's because of this peak that it creates this interesting modeling problem, because cognitive resilience, does it change linearly or not, is actually an open question. Again, the biggest factor is actually your educational attainment, still to this day. That's like every model keeps saying educational attainment is really important. But that's really weird because it happened so long ago. But education ends up influencing your work life, it influences who you marry. There's we can make a lot of hypotheses, but my original answer was around getting just more details of the brain, not just the imaging side, but some biomarkers. But now I've realized, or at least I hypothesized, that it actually might complicate some things. Whereas atrophy is done. Like, once you lose that section of the brain, you're not getting it back. So I don't know. It's, I guess, a little bit of the answer. I would love better cognitive data. That I think is very easy to argue for. But that's not the part anyone's fighting about. It's really about trying to understand brain health in people that are living. And for me, I mean, one of the things I'm doing right now is actually going to the autopsy studies and using the brains we have here at WashU, as well in other places, to really understand trajectories before their death. And then you have a really clean picture of the brain pathology, and maybe help that kind of inform what we're seeing in a living being. And you have all these measures that have some level of mismeasurement. And ultimately, I think our models are okay, but could they be more robust? Do they need to be more robust? I think this has been underappreciated about how complex it actually could be.
Thanks for talking through that.
Yes.
Thank you. That was wonderful. And I think I really appreciate the intervene early, let's get resilience as a public health approach. So I have a couple questions for you. I'm wondering if you could say a little bit more about how you created the cognitive resilience outcome marker. Was it sort of a PCA type approach? Were there factors that weighed in more heavily? And then my second question is, just out of curiosity, did you look at any iron metabolism pathways? So thinking about iron and its contribution to myelination, iron and its contribution and importance in inflammatory processes as well?
Mm-hmm. Just quickly on the second question, iron did pop up, and some other metals too, which I think were underappreciated in the nutrition literature. Selenium was one that was very strong. So in the paper, we discussed it a little bit more. There were quite a number of, when you think about what's intervenable, a lot of them ended up being diet related.
Selenium, I think, makes sense, actually.
Yeah,
Anti-inflammatory.
exactly.
Yeah.
So it was, yes, it did come out as an important factor and one that I think we could study a little bit more. As far as the measure goes, there's a lot of things that we tried, and then there's the one that we agreed to publish on, because ultimately, everyone knows that there's flaws with this approach, but no one has done so much better that the benefits outweigh the risk. But I'm trying, other people are trying. So at the end, we are just using residuals, actually. And it performs really well. And it's performed really well across studies too, which has been unexpected, but nice. I fully acknowledge that in the residual is everything you didn't measure, right? It just seems to be the case that at least a dominant part of that residual has some information that seems to be associated with what we're coming to. And again, that's why I used a theory-grounded approach to come up with a measure. Because if you're going to rely on something, at least have this framework that helps us understand if we're going off the rails or not. And so, yeah, for this model, it's just linear regression. We're taking the residuals out of it. I've done the methods work to show it in longitudinal models as well. We actually have more robust, I think, approaches with the longitudinal data, which is not surprising. And that work is also in the paper. But it works, and it's very easy to explain to our colleagues that are clinicians. And so for at least now, I think, until we come up with something better, that's what we're doing. Yes.
Okay, this was so clear. I really appreciate it. I'm a gerontological social worker, so we work on resilience, our concepts. So I love this conceptualization of resilience. But we're focusing on trying to get folks into cognitive stimulation therapy, into social engagement, into educational engagement later in life, we're looking at even internalized ageism and how it affects people. How does that fit into your thinking?
Yeah.
Because you focus beautifully on the biology of it all. What about the social part of this?
So part of the reason I love this work is because in my dissertation, I quote Descartes. I talk about sociology, psychology, psychiatry, neurology. It really allows me to kind of think about the brain, think about consciousness, think about knowledge and being, which is enough for a career. So I'm happy. One of the things I didn't go into today is about psychologic understandings of resilience and how they're associated with cognitive resilience. It's an area of work that I'm very excited about. I've proposed to do some of this work in the Jackson Heart Study, which is a study of African Americans living in Jackson, Mississippi, and they have really excellent data on things like optimism and other psychological traits that are of interest. There is some relationship. It's not very well established, but we see these signals that things that promote resilience from the psychological perspective are associated with cognitive resilience. But we don't know why. In some level, right, like the neuropsychiatric community, when they talk about resilience, we focus a lot, I think, on having multiple networks to represent a concept, right? And so the idea is like you lost some brain cells here, which was, encoding this one network, but you already had a backup network. And that's why you're more resilient, because, you just, through education, through overcoming adversity, whatever it may be, like your brain has come up with many ways to represent these things. And so it's okay if you lose a few ways. But some of those principles are also encoded in our psychological understanding of resilience. And it really comes back to this idea of like any sort of stress, is going to elicit a response. How are we as biological, psychological beings going to respond to it? So I don't know is the real answer to your question, but it's a very core question in this field. Like, again, we're using a very biologic definition, I think by necessity, because we're still struggling to understand and measure this. But when it comes to the intervention side of resilience, we're kind of assuming that it is almost not exactly mapping one-to-one, but very closely mapping to psychological resilience. That's why the interventions that people propose are things like cognitive training, things like optimism, addressing loneliness, otherwise increasing, engagement, thinking about retirement and how to live a meaningful life, these sorts of things. Yes.
So I'll ask the flip side of that question, which is to say that if we're going to medicalize, I should say medicalize dementia clearly is that fits within a concept of aging we may not want to medicalize. But it strikes me that with some of the data that you showed in the multi-omics, be that proteomics or metabolomics or even genomics, that we ought to be able to detect this disease much earlier than we do.
Mhm.
And now that there are disease-modifying agents, theoretically, with the beta amyloid-modifying drugs, you could, in theory, intervene quite a bit earlier. I have many thoughts about the medical side, but if you could maybe come in on the public health side of how we even start to think about that kind of sea change and how we understand dementia.
Yeah, so it's a very large debate right now in the Alzheimer's space around how we should define dementia. There's a very strong push from researchers in particular that dementia should move to a purely biologic definition based on amyloid and tau and a few other markers like NFL. And it is the opinion of mostly the psych side of the field that we have to hold fast to the clinical manifestation. And that has to be critical. And ultimately, both sides kind of agree, it's on that, but it's just how important are the biological elements to it. What this work shows, and again, this is not new, it's just kind of been underappreciated for the last century, is that just focusing on the biomarkers may be incorrect, or at least limiting. If we remember that these are humans at the end of the day, and what really matters is if I'm going to be able to function and keep my core identity, understand my sense of self, these sorts of things. So there's some parts of this that I think should happen anyway. Like part of it is around stigma. And the argument for the biological side is if we frame Alzheimer's as a chronic disease based on these biomarkers, just like we do with your cholesterol level or your blood pressure, it helps decrease the stigma. I agree that that is a very valuable thing to do because, my grandparents struggled with dementia, so it is scary to me, and it is, I think, very unfortunate that there are individuals that lose kind of their sense of self. But even that is not permanent. I think that's another in the weeds part of it is there are plenty of cases of end of life lucidity. People that have been demented for years just prior to death, then are completely lucid, completely themselves. And this is not like a one-off thing. Sometimes it's very extended. I've talked to a colleague who's a nurse researcher and researcher at Wisconsin, and she does some of this work. What does that say? If it was still kind of there, but unaccessible. These are just some of the things I think about, right? There's something going on biologically. It's very important. But for me, and this kind of work highlights it, the impact is not 100% certain. And even when you are dealing with the clinical manifestation, even that may be recoverable. Somehow, people are able to do it with kind of this end of life flush of neurotransmitters, and we don't really understand it. But the point is, like, some of that was still there. So from the public health lens, the first part of this is just promoting resilience writ large to avoid kind of these, hopefully to prevent individuals from going down this pathway for as long as possible. The other part of this from the public health standpoint that I think is important is when you look at people that are resilient, like some of our best cases of resilience, even if they get dementia, one of the fascinating things is like they will live, let's say they die at 75. They would live cognitively normal until 73, and then all of a sudden decline. But their time in dementia is actually really short. And we see this in familial AD, we see this in sporadic AD, and it comes back to this idea of health span, but also from the bigger picture, we think about the costs associated with it, the treatment associated with all the needs associated with that phase of dementia. If people were more resilient, even if we're just delaying dementia, they ultimately spend less time in dementia. And I think that from a public health standpoint has innumerable benefits. So yeah, there are definitely arguments for the biological side of it. I think the clinical side, where you're focusing on their actual cognitive function, should not be ignored, and we're not ready to discard that because we don't really know what the biology means yet.
Thank you all, and thank you.